Hi, class! Welcome to another episode of Andi's Chronicles of Biology. I'm here to help you understand anything that piques your curiosity as we learn constantly. Are you ready? I'm sure you are. For today's episode, we're going to be talking about the genetic blueprint of life. So, does it ring a bell yet? Well, yes, it's already on our slide. We are talking about deoxyribonucleic acid, or for what it's popularly known, DNA. What is DNA? DNA is the molecule of heredity. It transfers genetic information from parents to offspring. Just imagine the curly hair that you got from your dad, or perhaps the dark brown eyes that you got from your mom. That's all explained by genetics. And DNA plays a crucial role in these examples. Now let us explore the DNA structure. As you can see in the figure, DNA is a double stranded helix composed of small molecular units known as nucleotides. Try imagining a spiral staircase like this one. Are you visualizing it better? Well, I'm glad you are. Now, let's take a closer look at these nucleotides and see what it's actually made of. Nucleotides are made up of three major ingredients. In the sides of our double stranded helix, the 5 carbon sugar molecule, known as deoxyribose, and a phosphate group are bonded together. Inside this staircase like structure, 4 nitrogen bases can also be found. Introducing adenine, guanine, thymine, and cytosine. Just always remember, A likes to bond with T and C likes to bond with G. Quite easy to remember, right? Now let's move on to the replication. It's replication time. The DNA inside our body undergoes several important processes to ensure the growth and damage cell repair of organisms. But for today's chronicle, we'll only be focusing on level 1, which is DNA replication. It is essential that every cell is able to accurately duplicate itself. But how does this process actually work? Well, DNA replication is the process in which DNA is copied. For eukaryotic cells, this takes place within the nucleus during the synthesis phase of the cell cycle. On the other hand, for prokaryotic cells, this process takes place within the cytoplasm. Now let's meet the enzyme family. For today's class, we'll be playing an amazing 3D animation of DNA replication that is provided by HMMI Bio Interactive. The replication starts once the helicase unwinds the bonds between the complementary bases in DNA. During this process, single stranded binding proteins ensure that the broken bonds does not attach back together. During this process, the leading and lagging strands are formed. On the other hand, topoisomerase plays a crucial role in preventing DNA supercoiling. Once these bases are exposed, the primase does its job by adding the RNA primer into the leading strand, which will then be recognized by the DNA polymerase, initiating its addition of new nucleotides to the strand. The gaps present in the Okazaki fragments formed in the lagging strand needs to be joined together. So this sounds like a job for the ligase. This enzyme is the one that fills the gaps and glues them together. Fun fact: DNA replication is a semi conservative process as it contains one old strand from the parents and one new strand built by the polymerase. In this process, we can clearly see in the 3D animation how does the DNA replication actually work and how does each of these enzymes that are part of the DNA replication actually does its job. Wow, that's surely a lot of information. I'll be introducing more about these important DNA processes in another episode. For now, that's it for Andy's Chronicles of Biology. Here are the references for this video. See you again on another episode. Thank you for watching!